Okay, just to end off, I'll see how much you've learned, understand the system. You can give me a score. There's no answer in these. MS would be good enough, I think. MR. There's the one with the nodes. MS. MSS, but you have to say just the node. You've got to qualify the S part because it's not the entire stem. Easy? R. Easy. MR. MR, in some cases it could be MR, MS probably. MS, good. So these were the pictures we've actually looked at now in the session. Now for some new ones. MR would be a little bit high, I would think. MR, MS maybe. And again, quite a bit of leaf rust on there. But you have stem rust. MR would be fine, I think, yeah. MS? MR, MS probably. R? MR, MS? MS? MR, MS probably. According to my system, but we know we all have a different view probably. VS. Yeah, you can also get a VS. You can't see it on the screen, but if you come have a look on the computer, that is mycelium of a contaminant. And the reason you see it there is I cut these stems in the field, and I put it in a, which I shouldn't say aloud, a plastic container. But I did not cover it. I had my sandwiches in there on the way to the field and then brought it back in the same. I just put a piece of wet paper towel in the bottom and then the stems on top. I didn't cover it. And I took the pictures the next day. But even within 24 hours, and they weren't covered completely, you can see the contaminants growing. I think that's the point Dave made. Never use plastic bags to send the samples. Make sure they dry when they go in. Because you get all these unwanted organisms that will interfere with the DNA analysis in the end. Brown rust. First prize. That's not stem rust. But usually in our nurseries we have all three, so it's very difficult to separate. Just when you take the notes, make sure that that's not stem rust. Well, you can see the telia, and at that point we didn't have any stem rust telia. So. No. And you can see in the leaves, all the leaves are dead, so you assume that the leaf rust was pretty high on that particular entry. Yeah, that's probably MS going to S. But again, it's not like we see in the textbooks. You see all kinds of variations. because these are polycyclic diseases and some infection sites will be younger than others. So you get the variation and you must, take that into account. I think we're almost done. Lower severity, but with fairly large pustules. Probably MS. You can maybe even call it 5S or something if you want. Easy. Resistant. So you can see where the problems come in the gray areas. The extremes are easy. It's the M category. Again, towards the nodes. Huh? To make an N note in your field book. MRMS probably. And that's it. Thank you very much.